In this video, we're going to take a look at some of the diversity of microscopes that are out there and the kind of microscopy that you can do with these sorts of scopes. So we'll start by looking at light microscopes. So the work that we're going to be doing is referred to as bright field microscopy, most of what we'll do at least. And it's the type of microscopy where you see a dark object against a light-colored background, what is thought of as the bright field. This background area, this is the bright field. So it's a light color background, and you can see darker objects, being magnified against that light background. And with bright field microscopy, we're transmitting light through the specimen, and we can even stain that specimen to make the structures darker, to increase the contrast with the background. Although that staining process generally kills the specimen. So in order to really get a lot out of bright field microscopy, we stain, and unfortunately, or I don't know, not unfortunately, it doesn't matter. This is going to kill the microbes that we're looking at. So that's bright field. It's also possible to do something called dark field microscopy, in which you use a special condenser, and then that transmits light through the specimen. It prevents transmitting light through the specimen, and instead the light reflects off of the specimen at an angle, and it makes the specimen brighter and the background or the field darker. So that's a dark field microscopy. So that requires some special equipment to equip your light microscope with. Another type of light microscopy is called phase contrast microscopy. And there you're using a specialized condenser again and lenses that really take advantage of the refractive indices and the differences in the refractive indices of the internal structures. And this allows you then to observe live specimens because there's no killing stains required. So this is a means of really enhancing or taking advantage of the differences in refractive indices so that you don't have to kill the specimen. And you can see things like motility, their ability to move. That's motility is that term that I just did out there. And so with phase contrast microscopy, because you haven't killed the cells, they can still move around. And if they've got flagella that move them around, then you can actually see some of that movement. Another kind of light microscope, and again, this is not any kind of microscopy that we're going to be able to use, is called differential interference contrast microscopy. And it relies on the differences in refractive indices to, of the specimen and the stuff that's inside the specimen. But it produces a kind of almost 3D image. The image is so contrasty, and you can really get a lot of detail using one of these differential interference contrast microscopes. Fluorescence microscopy also takes advantage of fluorescence. Remember from the previous lecture, fluorescence is when light energy is briefly stored in an electron, and as that electron loses that energy, then it releases longer energy wavelengths of light. And so you use UV light with fluorescence microscopy to bring up to produce glows around those molecules that are going to be able to fluoresce. And so you see these brilliant orange, yellows, and yellow-green colors based on these fluorescing molecules. And if the specimen doesn't naturally fluoresce, then you can use fluorescent dyes. So stain it with a fluorescent dye to accentuate the parts that you want to look at, and then use the fluorescent light. And we can even use antibodies, which are, we'll talk in this class about antibodies toward the very end of the class, but these are specialized proteins that can go and bind to very specific proteins. So you can build antibodies to bind to particular proteins and carry along with them fluorescent dyes. And so this can be used for rapid diagnosis if you're trying to see whether there are specific proteins, marker proteins, on cells. So all of this is under that umbrella of fluorescence microscopy. So Nomarski and fluorescence microscopy, and then confocal microscopy. And this time you're using laser light that is delivered through optical fibers that illuminate the dyed specimen. And then you take a series of digital images as you focus through different zones of the specimen. And so it's like taking these little thin sliced pictures, digital pictures of the specimen, and then using some fairly complex computer programs, the computer can reassemble these individual digital images into a three-dimensional model of the specimen. And you can also use it to do time-lapse images of living specimens as well. So you can watch a biofilm grow or living specimens move around using confocal microscopy. So, and I'm showing you there two images then, one using just a standard fluorescence microscope and one using a confocal microscope. And you can see how much more detail the confocal image gives. And then last of all, with many of these microscopes, we can now produce digital images that can then be manipulated through the computer, although you cannot achieve greater resolution. So all you can do is do digital magnification through the computer. You're not going to be able to increase resolution on those digitally magnified images. But it does allow then false coloring later on so that you can produce images where you highlight specific structures as well. So it just provides the chance to then work with that image to be able to measure things or digitally or to highlight specific structures. So light microscopy is very limited. As we've said, you can only resolve objects that are larger than or separated by 0.2 microns. So that doesn't give you as much resolution as we might wish to have. Because the subcellular and intracellular structures just cannot be fully resolved using light microscopy. They're too small. And something like a virus is essentially invisible. There are maybe a couple of viruses that are large enough to see using a light microscope, but for most viruses, they're just not going to be even visible using a light microscope. They're way too small. So that's when we turn to tools like the electron microscope. So rather than using light, electron microscopes focus a beam of electrons on their subject, and rather than focusing them with glass lenses like we do in light microscopy, you use magnets because electrons have a negative charge, and magnets can be used to focus them, to force them to cluster together, even though they have negative charges that are working against each other, that clustering. So we focus this beam of electrons onto a specimen that is then maintained in a vacuum. You have to evacuate all the air because the air molecules would cause the electrons to scatter, and you would get no image at all. You just see a lot of blurry fuzz. But if you in order to put that specimen into a vacuum, then it has to be killed. So that if nothing else, the putting it into a vacuum would kill it. but the preparation processes required to be able to visualize using a light microscope also require processes that won't allow the organisms to live. But using electron microscopy, we can get super high resolution magnification. And so, and what we produce from electron microscopy is referred to as an electron micrograph. That's the image. And it's either an electronic file or you can then print it out as a picture. So, and here is an example of an electron microscope that's it's currently being used at the University of South Carolina as part of their tools that the biologists there use. So there are a couple of different ways that electron microscopes work. One is using a process called transmission electron microscopy. And in this case, whatever the structure is, so let's say you want to look at a piece of liver tissue, you would cut off a small chunk of the tissue that you're looking at, or the tumor that you might be wanting to look at, and preserve it, and then encase it in either wax or a relatively soft plastic. So you get this cube of material that has the specimen embedded in it. So there's our specimen inside the cube. And then using a special knife, it's often done just with a piece of glass that has been freshly broken, so that you get this super sharp edge. Or they use diamond knives as well. So again, diamonds that have been fractured, but they're super hard. The glass, every time you're setting up a microtome, you would make a new glass blade where a diamond knife would last longer. But you then shave off thin slices. It's almost like shaving cheese off of a block of cheese. And then you collect these shavings, and you find one, you look at it under a light microscope first to find one that has what you're looking for in it. And then you can place that little shaving into the electron microscope. And now, so we have this shave of wax with a thin slice of whatever the material is. And then electrons pass through it, either from, they're probably coming from the top down. So electrons pass through it, and then at the back side of the electron microscope is a collector. And so some of the electrons are going to be absorbed by the material that's in that shaved wax. And some of them will pass through, and similar to the way X-rays will pass through material, but some of them get blocked. You have the electrons passing through, some of them get blocked or absorbed, and that produces an image that is after having been transmitted through that material. So the sections have to be super thin because electrons cannot penetrate very deeply. And they can also treat the sections with heavy metals that will increase the electron scattering so that you can really enhance the image. So that it's possible to enhance the image by treating that section with some metals. And with TEM, or transmission electron microscopy, we can produce images that are magnified 500,000 times, and then you can further enlarge that using the computer up to 20 million times, and it has the capacity to resolve structures that are 1 nanometer apart. So that's the same as 0.001 micrometers. So that is much better than the 0.2 microns that the light microscope allows us. We get significantly greater resolution. So that's transmission electron microscope, where we're sending the electrons through the particle, through the thin slice of the material that we want to look at. There's also scanning electron microscopy, and here, rather than transmitting the electrons through, here we're trying to get a sense of what the surface of a specimen looks like. And so we coat that, you take a killed specimen, coat it with a heavy metal like gold or palladium, and then you pass this very, very narrow beam of electrons across the specimen. And so as this beam kind of moves over the specimen, so if this is our specimen here, well, here's our specimen. So we are taking a picture as we pass this beam of electrons across here, and we're collecting the electrons that get scattered back toward the receptors. So those reflected electrons are what make the image. And so you see across the screen this image as the electron beam passes across the structure. And so you can watch that as you move around. You can watch the structure as it moves underneath the beam of electrons. And you don't get quite as significant magnification here. Only 50,000 times magnification, and you can then further enlarge it, a reasonable amount digitally, and it resolves structures as close as 20 nanometers. And so if we took 20 nanometers and turned that into micrometers, that's 0.02 micrometers. And again, remember light microscopy, it's 0.2 micrometers. So you get about 10 times the resolution that we can get from light microscopy. And then there are a couple of other very high-end, high-tech microscopes, and they include scanning tunneling microscope that uses a super thin wire probe, and there's an electron cloud surrounding the metals of that wire probe, and that electron cloud, as it changes shape, as that probe kind of makes its way, fingers the specimen, then it tells us about the shape of the specimen at the molecular level. So we're looking at the shape of the atoms in the molecules. And because STM works underwater, it can be used with live specimens, if you can pin those live specimens down, if they're not moving around. And then atomic force microscopy can get all the way down to images at the atomic level, so we can see images of atoms all the way up to 0.1 micron. So it can take super small to very large images, or produce them to almost the same kind of resolution as light microscopy. So you can get a real range from the angstrom level up to 0.1 microns. And it can distinguish among the different nitrogenous bases, so adenine, guanine, cytosine, and thymine. It can distinguish between the 4 different bases that are found in DNA. It can watch the changes in molecular structure as chemical reactions occur. And it can even move atoms around and measure the force required to do that movement of those atoms as we deform molecules. So atomic force microscopy, again, really high-end, high-tech type of microscopy. Okay, so there you have it. Light microscopy and the different types of light microscopy and their uses, as well as electron microscopy and the uses of these tools as well. So, as always, you know what to do if you need to reach me. Phone call to my office, email, probably the best way, or even better, actually, for face-to-face is come see me during office.